Yes, so Morena, Tena Kote and thank you very much to the organizers for this very inspirational conference. And I couldn't have had a better introduction, Philippa. Thank you so much. I can actually skip quite a few of my introduction slides, I think. So I don't need to introduce you to the circadian field anymore, and Mario did this morning as well. So I'm also a hobby astronomer, a very bad astrophotographer. That was my first picture, although I do hobby astronomy for 30 years, but I only do visual observing. And this was my first picture I just took 2 months ago. But it illustrates how dark it is where I live, which is in Dunedin in the South Island. So we are very lucky to be here. Well, let's see if that works. So, as we heard already, we evolved through the solar cycle, the rotation of Earth, and our physiology in our body has evolved to this. And
you're checking the wrong way.
Yes, I just got it from PowerPoint, I must say. So, for example, at around 10 o'clock, 11 o'clock, you have your highest alertness, which would be now, so we have a good timing for our talks here. But it's not after the conference dinner, after stargazing last night, which the speaker did, me, so you will be with me, please, if I fall asleep. No. So, our master clock resides in the hypothalamus, and this region is the region that I'm most interested in. I'm a neuroscientist, and this region does not only control our circadian physiology, it also controls metabolism. So there's some overlap, and I'll talk about metabolism in my talk. It also coordinates the peripheral clocks that we find in other organs, for example in the liver and the heart and so on. If you get disruptions to this circadian rhythm, as you already heard, we get associations with mental health and mood disorders, cancer, cardiovascular disease and metabolic disease, just to name a few. And I would like to focus mainly on metabolic disease during my talk. Well, we are lucky in this field that it's now very well recognized by the award of the Nobel Prize in 2017. And this was awarded for the machinery. So how does this actual clock work? So if you consider we actually have a clock in the brain, it's really hard to understand this thing. This watch might be in your brain. And well, it's actually certain genes and proteins that are expressed rhythmically and that then they can inhibit each other. It's a rhythmic cycle of gene expression and protein expression. And this is how the clock works. And it was discovered in fruit flies, so a lot of our knowledge in medicine now comes from fruit flies, and this is what these 3 people did, and they received the Nobel Prize. The first gene that was discovered was period, so that's one core clock gene, and I'll come back to that later. So why did I get interested in circadian physiology? So I need to go back to my PhD, and I worked on a very fascinating animal model, and this is the Siberian hamster. It lives in Siberia in an area called Jungaria. And what it does, it changes its body weight just by light. So under 16 hours of light and 8 hours of darkness, it weighs around 40 grams. Then you give more darkness, so you give it 16 hours of darkness and 8 hours of light, more like what happens in winter, and it gradually loses weight, about a third to 40% after 3 months it's fully adapted. And my supervisor said, find out why it changes the body weight. So it was a big task, and it took me more than 20 years, but I come back to that later. So, and I got sidetracked, I must say. It's my main topic, metabolism research, so I was focused on this. But when I did this experiment, I didn't take into account that somebody came into the university. Oops, pressed the wrong button. So it stopped working again. Can we please press? So this guy came, the security guy, opened the door to the facility. There was a sign, don't open, it was locked, but he needed to check that no burglars there stealing my hamsters. So the light came in just from opening the door for a few seconds. The hamsters thought, okay, let's increase the body weight. So can you press again, please?
Still not working.
No, it's not working. Or can we change the battery?
We have batteries.
That's not the battery? Okay. So they went back to normal body weight, to the long 40-period body weight. So I couldn't do the study anymore, so I had to start all over again. So that you remember. And that made me interested in circadian physiology. But as I said, I'm a metabolism researcher, and we worked on something different. And we worked on the hormone leptin. And my supervisor discovered the receptor of this hormone in the brain. It was discovered in 1994. And these mice were around for a long time in the laboratory. And there was a hospital in Cambridge where they had children that were obese. And and these children had a lack of leptin, and the mouse as well, so you could inject them with leptin and they lost body weight. Amazing. So we could cure the obesity pandemic, but it's not that easy actually, because leptin is produced by adipose tissue, by fat cells. When we get obese, we get more fat, and the brain doesn't read the signal anymore, it becomes resistant, the hypothalamus region in the brain. It normally reduces food intake, increases energy expenditure, and it regulates blood sugar. And this is really important for the talk. I'll come back to that later because I focus my main research on that. As we get obese, we eat the wrong diet, the Western style diet, we become resistant, and the hormone doesn't work anymore. So we cannot treat these humans with leptin, and 95% of humans, obese humans, are leptin resistant actually, so it doesn't really help us to treat them with the hormone. So when I moved to New Zealand in 2014, I went to the observatory and people said, you need to fight light pollution. But I worked on leptin for the last 10 years, and I thought, maybe I can leave a little small impact or some little studies that could help the field. We can do this in the lab, and I thought I need to come back to circadian research. And what we did, we used mice, and we didn't travel with them around the world, but we gave them 12 jet lags, as if they were traveling around the world every 6 days in a 6-hour time shift going east. And what you can see here, Nathan Skinner, the PhD student did the study, you can see that the body weight increases. Also, the resistance, the brain became resistant to the hormone leptin. We discovered that as well. Well, we looked at the regulation of blood sugar, and 100 is the normal range, below 100, and here you see they have a massive dysregulation of blood sugar. The levels were 3 times higher, which was very striking. I had another PhD student, Alisa Buxheim, and we thought we need to go back to this dogma in physiology. So when we get obese, we are leptin resistant. That was always what we are told. But maybe this is controlled by the circadian rhythm, and interestingly, nobody looked at that before. So here we have mice on a normal diet and mice on a high-fat diet. And when you feed them a high-fat diet, people do the experiment in the morning. Everybody in the world normally gets up early, goes to the lab, and you see the response is reduced. This is the leptin sensitivity at a molecular level. It's reduced, they're leptin resistant. But actually in the evening, when nobody does the experiment, when the light turns off, they are sensitive. So that was very interesting. We came up with this model that we should potentially eat during times when we are leptin sensitive and not when we are resistant. And this was very topical at the time because time-restricted eating came up, so that we only eat during a certain time of the day at regular times, as food, as we already learned, is another input into the clock to reset the clock. Well, that was 2019. I presented this at the conference, and then something happened, obviously. COVID came. And we couldn't continue, so we had another disruption in the lab. And I had to focus on the core business, and this is to find something to reinstate dysfunctional hormonal balance in the brain that is the root cause, considered as the root cause of the development of diabetes. And I'm one of the very lucky researchers who met a couple in Invercargill who happened to grow dahlias, and this is just a bit of sidetracked, but it has some relevance here. I'll just mention it briefly. So we found compounds in these flowers that can reinstate the hypothalamic signaling of the hormone leptin and insulin in diabetes, and we discovered that in animal models and confirmed that in humans. and now it's grown commercially in Christchurch at a larger scale. And it has been picked up by international media, so we might now be one of the first to target the brain as the root cause for this disease, potentially. And the journal made this cover art. So we developed this brand. It's a natural supplement. With supplements you cannot make any claims, but it's fully made in New Zealand and comes from the Behaviour flower. So why do I mention it? Well, this was the dream come true for me. It's very exciting to me and my lab, but it has also relevance here because chronomedicine is important here as well. When we look about medication to regulate blood sugar, this is an emerging field. You take the medication in alignment with your circadian rhythm. And we designed this clock when you should take our supplement. And what else you can do when sleep, you can find there as well. Block light out of your bedroom, for example. So that was 20 years of research summarized, so that was my main research that led to this. But I thought I need to come back to the circadian regulation, and COVID is now over, and we, the conference approach, so I thought I need to come up with some new data, because I want to come back. And we now use a different animal model, a zebrafish, which is quite interesting because it is actually diurnal, like humans are, whereas the mouse is nocturnal, so that's an advantage. Another advantage is you can look through it in early development. It's transparent. You can see what happens in the fish. Then I was lucky again, and Pramuk Keartisinghe came to the lab, who came from Auckland, who was a zebrafish expert, and together with his supervisor at the time, Chris Hall in Auckland, they asked the question, does the circadian clock control immunity? And we heard something about this already this morning, but they wanted to know if it changes the response to infection, clearance of infection. So what they did here, they took a part out of the clock, so the clock doesn't work, the period gene, they mutated that. And then they had fish on a normal light cycle, constant light, a reverse light cycle, and constant darkness. And they infected them with salmonella that were actually injected into the hindbrain of these fish, and you can see them in green. So what was surprising, what they found, or I find that a striking experiment. So normally 50% of the fish die, so this is the curve where you can see how the fish die. And so 50% of them die, but if you knock out the clock, if you mutate the clock, 75% die. And this was dependent on the light period. And if they do a rescue experiment, we do this in biology, where we bring back period, then the effect is gone and only 50% die again. So there's a link to the clock, and on this slide it becomes even more obvious. So here you can see the bacteria in green. So they get, after 24 hours after the infection, they get these the staining. And they spread rapidly when the clock is mutated, and this is just the quantification of it. So I thought, what can I contribute to this conference? And we had remaining questions. Now I talked about circadian rhythm and light, and light disruption, but which light color temperature is important? And we came back to blue light. We wanted to know if it affects behavior, growth, and metabolism in zebrafish. And right here, a new honor student came on board. People need to climb mountains in my lab because we are the metabolic lab, and he was happy when I told him about this project. We bought a machine, the university bought it for us, which was very expensive, which could actually now measure the behavior of 96 fish at the same time, which was amazing. And it came with these 5 different light sources. So we could illuminate the fish with white, blue, or no light, blue light, white light, green light, and red light. And we wanted to know, how does that affect the behavior first? So after 5 days of this exposure for 12 hours, day and night, so we only gave the light during the day, we then measured the behavior. And what we can see is that the green and red light leads to a lot of swimming behavior. They move a lot. But during, when you expose with blue light, they don't move at all, as if you give no light. There's no difference. White light is somewhere in between, but remember there's a blue peak in the white light that could explain what is in between. And this is just showing the statistical analysis of what I just showed you before. It's not so important. Then the student came and said, I think the fish are shorter. And I said, really? Let's measure that. So he measured the length of the fish, and you can see that blue light exposed fish are shorter than fish exposed to the other two light sources. We didn't do the red light yet, but you see that they are shorter, about 7 to 8% compared to the white and green light. Since we are metabolic lab, I thought, oh, we need to measure metabolism. So we looked at the glucose metabolism, and we found something really striking. Only blue light increases the glucose metabolism, which we don't understand yet. But the other lights had no effect on that. So it seems like development is impaired in that fish, metabolism is increased. Increased metabolism is often associated with aging. And we missed the leading study that shows that light at night, for example, can increase mortality in humans. And Sean came here 5 years ago, and luckily enough, he listened to our calls, and he published this last week. It actually came out today in the published journal, I think. And what we showed in 88,000 individuals, so this is a really good study, that brighter nights and darker days predict higher mortality risk, and this is just the highest percentile of light intervention, and you see that if you give that during the night, you have a higher risk of all-cause, cardiometabolic, and other-cause mortality. So to summarize, what did we learn? Well, maybe it's all about aging. So we can talk about all these diseases that occur when we do circadian disruption, and metabolism is very strongly linked to aging. So maybe it's all about aging. And as we age, now even the Bug Institute, one of the leading institute for aging research, picked this up as one of the leading factors of aging, circadian rhythm disruption. And they could also show that this rhythm of the amplitude of our circadian rhythm goes down as we age. In waking activity, the SCN firing, for example, cortisol levels, melatonin, plasma glucose, and so on. I don't want to say when it's a flat line, what happens then. Well, who wants to live longer here? That's not many. Who was the lighting engineer? Who was the politician? So I think this might be a topic that unifies us all because we're all interested in this. And if we want to live a healthy, long life, it may be beneficial. Some take-home messages to avoid bright light at night, particularly blue light, both in and outdoors. It's equally important to seek bright light exposure during the day, which we didn't talk about, but this is very important to suppress melatonin during the day and reset the clock also through the photic stimulus into the SCN. Eat meals at regular times, very important to reset your clock with food, and try time-restricted eating, which seems to help for improving our metabolism, and thereby also improve aging. Avoid frequent jet lag if you can, as we learned from the mice, or shift work, as we very well learned, but it's hard to avoid this. But there are ways by adjusting your food, your timing of eating, for example, and timing of lighting, where you can adjust to these. And live in alignment with your chronotype. Some of you have a shorter rhythm, some a longer rhythm. Find out what your rhythm is and try to find a job that suits that rhythm, if you can. Well, this is all from me, and I would like to thank all the people involved in the labs in the different countries, my German lab at the time, and the funding bodies. Thank you very much.
Excellent talk. I really appreciate that. I'm curious about that extract, dahlia, that you mentioned.
Yeah.
I never heard of that one before. How does that work biochemically? Is it working in the pineal gland or peripheral tissues? Can you just elaborate a little on that?
Oh yeah. So we showed that it acts on the brain in the hypothalamus and then reinstate insulin signaling that is disturbed during obesity. So we lose insulin sensitivity, which is caused by leptin sensitivity. That's too complex for this audience. But no, sorry. I didn't mean that, no.
time you've got available.
Hello. Sorry. I didn't want to say it. No, I mean, we're so diverse, I can't go into the details. So, yeah.
There was one more.
Okay. I just wonder, like, humans are dispersed along different latitudes at the on the world, and I just wonder, like, the day-night cycles are different if you, for example, live here compared to the equator or live in high latitudes. So I just wonder whether you can see whether there's a study that looks on the different, which impact that has, because when you, for example, live here, you have like, it's all different than in Europe, but I'm confused, but like longer days during summer and shorter days during winter, so.
Yeah, we had, sorry, that's a very good question. We had this discussion. I'm not aware of a study directly, but I just recall something, I don't know if it's true, we need to look at that. But if you have less input into your circadian rhythm by constant light and constant darkness, if you live without any photic stimuli, close to the pole, for example, then your circadian rhythm would be dampened, and that should lead to higher mortality. I need to look at that study. I think there is some correlation with the latitude and longevity, for example, but I'm not 100% aware of that. So, very good question.
All right, we're going to have to move on, but we have one very quick question here.
Oh, yeah.
How do we find out our own cycle?
Your own cycle, go back to the lockdowns. I found that my own cycle only then. I always thought I'm a lark, but I'm actually not. So when you woke up there, that's the time you should, if you can, carry on working or waking up. You know, if you wake up at 7, wake up at 7 and
There you
go
go,
at 6.
you've got an exercise to take away and try it. Now we'll move on. Thank you very much, Alexander.
Thank you.